Good evening all of you. Welcome back to our botany session. Today's topic will be cell, the unit of life. I hope so everything fine. I am audible to everyone. Board is clear to everyone. Give me the response please. Okay, very nice. So in our today's topic, what we have to go with about the cellular biology, right? The topic I already mentioned you, cell, the unit of life, right? To enter into today's topic, first of all you must know about one interesting thing which you call it as hierarchy of organization. What do you mean by hierarchy of organization? Hierarchy means from lower to higher or higher to lower. Fine. So here organization means how organisms are going to be formed. Basic unit we are going to start with the cell. Last time we discussed the tissues level and before that we discussed the morphology. So today we have to discuss this level which is a basic unit to build an organism. So in hierarchy cell is at the basic level and cells are constituting tissues that we have already discussed last time. Then tissue, different tissues constituting what thing? Organ. Organ is forming what thing? Organ system, right? An organ system is forming what? An organism. If I am putting the arrows like this from cell towards the organism means I am moving from lower to higher level. But if I am keeping the arrows like this means we are moving to lower level from higher. Are you getting this point? Very nice. So we all are listening. Please don't chat with each other in this room now. I can check what you are doing here in the chat box. Please do focus on our main topic, right? When you will come here, that's you are having lot of time to discuss with you, but right now fully focused on this topic. So I repeat, hierarchy means something from higher to lower or lower to higher. So we are taking hierarchy of organization. Hierarchy can be of anything, but right now we are taking for organization from the cell up to the organism. And which level today we are picking? The cell. Tissues we have already discussed last time and organism level even we have discussed in behalf of the morphology of the flowering plants, right? Now let's start with the topic, the cell. There you know very well, by this can I say cell is the structural and the functional unit of life? Cell is a structural and functional unit of life. What do you mean by structural? Means an organism has to be formed, it has to start from the cell and it is going to be the functional unit. If cell is working all over, the cell when it is working, it is performing all the function almost which can organism can perform. If I say unicellular organism, hardly it is made up of one cell. It is performing all the functions. So cell is the basic, means it is going to be the base of the organization which has to collect together to form tissues, tissues into organ, organ into organ system, then into an organism. So cell is the structural and functional unit of life. We cannot imagine life from without the cell. It has to start basically from a cell. Now if I discuss a little bit about the history, the scientist related to that is a Robert Hooke, who discovered the first cell which were the dead cells, and from where he took the sample, from which part of the plant? From the cork part. What is cork? Cork is the dead cells which are on the outer side of the woody plants. Like, have you seen in your chemistry labs, the glass bottle is having some wooden type of the block at the mouth, right? That you can say as a cork, which are dead cell and taken from the part of the plant. Just keep in your mind for a while that who discovered the first cell? Robert Hooke. And from which part of the plant? Cork. But the cork cells are which cell? Dead cell. So can I say first discovered cells are the dead cells? And who discovered that? Robert Hooke. Are you getting that? So here we are discussing just the beginning of the topic, cell, the unit of life. A small introduction to that. Obviously what we are discussing we will again explain in the class. Here only we are bridging you from your 10th standard to the intermediate, right? Don't think that your +1 syllabus is going on, right? This is simply a touch of a +1 by attaching to your previous classes, right? Okay. Now next what we have to take, if I discuss regarding this point, that cells types, how many types we can take, that types will decide which type of organisms we are having. Like if I am taking unicellular organisms and the multicellular organisms, based on, I must say not based on the types of the cell, I must say the types of organism, the better word is types of organisms based on the number of cells. Now can you split the term? Uni means what? One. Cellular means what thing? Cell. The organisms which are made up of only one cell, that you call it as unicellular organism. Multi means what thing? Many. The organism which are made up of many cells, that you call it as multicellular organisms, right? So here I repeat, we are discussing about the cell, and based on the number of cell, two types of organism we discussed, unicellular and multicellular. Can I say the example of unicellular will be, for example, like bacteria is an example. Can I say amoeba? This is unicellular. Many more we will take. Multicellular, for example, like plants. Which plants specially? I am discussing higher plants. For example, this is all like an example. In the lower even we are mostly having multicellular. Even unicellular are there in or you can say lower type of the plants. Now even the animals which are multicellular, right? So based on the number of cell, two types of organisms are there. Now coming to the next point, we are having in comparison with each other different type of the cells. Like if I say based on the comparison of their sizes, like based on the sizes, we are having different types of cell. Even based on the shapes we have different types of cells. Based on the activities we are having different types of cells. Fine. First of all we are taking based on the different types of sizes, right? Now here see this diagram. In this diagram you will find that this cell is a eukaryotic cell. What is eukaryotic cell? I will say after this discussion, right? Yes, humans are multicellular, right? Which are having only one cell, that you call it as unicellular. Human, any complex organism, mostly they are what? Multicellular. Are you getting that? We will discuss more classification, right? In that we will discuss the various kingdoms. Maybe you have learned about Whittaker system of classification, where 5 kingdoms we already discussed, right? Maybe you have discussed already in 10th standard or wherever. We will discuss back again when you will come. So there the more classification will be clear to you people. So eukaryotic I will discuss later on, but comparison of eukaryotic, which we take the bigger in size as compared to other cell. like typical bacteria. Typical bacteria means which is having almost all the feature. We are assuming that you call it as what thing? Typical bacteria. What is the size of that? 1 to 2 micrometer. Then for PPLO, what is PPLO? That you call it as pneumonia like organism. Pneumonia like organism. What is measurement? That is about 0.1 micrometer. Even this PPLO, some books are referring that is a mycoplasma, right? And they are referring it 0.3 micrometer, right? So we can say one and the both same thing, pneumonia like organism and mycoplasma. These are the type of the prokaryotic cell. Now what is eukaryotic, prokaryotic, still we have to discuss. Just focus on the different sizes of the cells we are having all around, right? So we are having this much of a bigger whose measurement is 10 to 20 micrometer, right? And the smallest one is PPLO, which you even call it as mycoplasma. Its minimum value is 0.1 micrometer and some books are given it 0.3 micrometer. Are you getting that? Now coming to the next part, if I take a typical bacterial cell which is a prokaryote, what is the measurement of this? 1 to 2 micrometer. Now please focus everyone over here. Here viruses, can you see here measurement is still very less, 0.02 to 0.2 micrometer, even less than mycoplasma, right? But still we say mycoplasma is going to be the smallest cell. Why not the viruses? Because viruses are dead outside the body of host. What do you mean by host? Where they are putting infection, like human beings, right? When they come in contact with plants, animals or human beings, they become living. Otherwise as such in the air it will be dead. So we cannot say them as a living organism, right? So although they are smallest in its dimension, we cannot say they are smallest in their dimensions. We will take smallest living cell is what thing? Mycoplasma. Even you call it as what thing? Please repeat that if you are listening me. What is the other name of PPLO? Mycoplasma. Are you getting that? So right. So here the other name of PPLO is what thing? Mycoplasma. So we have slightly different measurement of it. According to one paragraph, when you open your +1 NCERT book, in paragraph they have written 0.3 micrometer. But when you see this actual picture there in your book, they have mentioned PPLO measurement 0.1 micrometer. But PPLO and mycoplasma one and the same thing. Are you getting that? Very nice. You are responding. That's more sufficient for me. Your focus should be here, right? Okay, so this is all about what thing? Different sizes of the cells. Now coming to the next part. If we are discussing the cells based on their shapes, we will have variety of shapes of the cells. See here, if I am taking red blood cells, you know that in our blood we are having RBC. What is the full form of RBC? Red blood cell. What is the shape of that? Round and biconcave. This is the actually shape of the cell. White blood cell is amoeboid. Amoeboid. Oid means like. They are amoeba-like. Amoeba is irregular in shape, na? Like this. So they are amoeboid in shape. They don't have such fixed shape. That's why you call it as amoeboid in shape. Now coming to the columnar epithelial cells. These are in case of animals you will learn. That is having long and narrow type of the shape. Can you see different shapes are there in front of you? Now coming to the next thing. What is that? This is going to be the nerve cell, which is the branched and the long, right? And can you see tracheate? We discussed already in anatomy. Can you say me this tracheate is an element of which complex permanent tissue? We discussed in our last lecture. Everyone, tracheate is a part of which complex permanent tissue? Very nice. Very nice. So here tracheids are the element of what thing? Xylem. One of that. We discussed vessel and tracheids, right? So this is one of the member of the xylem, tracheid. Now mesophyll cell. This you will find we discussed already in leaves, but we didn't discuss that in much. But we discussed mesophyll cells in the leaves. What is the shape of that? Round and oval. Whereas tracheid is elongated. On that day even we discussed it is tapering and broader at the center. So can you see the different types of the shapes are there around us for the cells and even for the sizes. Clear? Now if I say for activity, even their activity varies from place to place, right? That we will discuss in the respective topics about their activities, right? Now coming to the next part. So just now I discussed with you, we will take this concept. What is the difference between prokaryotic and eukaryotic cell? What is a prokaryotic cell? Where there is no membrane around this nucleus. Keep in your mind, like our body is under the control of a brain. In the same way, a cell functions are in a control of the nucleus. Nucleus is known as a brain of the cell, or you can call it as a controller of the cell or boss of the cell, right? So that nucleus is not well defined in the prokaryotic cell. Pro means what thing? Primitive. Pro means what thing? Primitive. Here karyon means what thing? Nucleus. If the primitive type of nucleus is there, that type of cell you call it as prokaryotic cell, or that organism you call it as prokaryotic organism. Getting that? So what is a prokaryotic organism or a cell which is not having this membrane bound nucleus? What is membrane bound nucleus? Can you see this is an animal cell, inside is a proper well defined nucleus is there, right? So that nucleus is not going to be well developed in case of prokaryotes. And moreover, they don't have membrane bound organelles. Which organelles we will discuss in the chapter. But at least you should focus that organelles are not compartmentalized. Suppose a building is there, a school is running, the proper classrooms are there, right? Every subject is running in their respective time in their respective classes. So that is the compartmentalization. But imagine a school where everyone is sitting in a one room of different, you can say ages and of for different subjects, right? A little bit chaos can be there. The things cannot be managed properly, right? So that's why it need to be compartmentalized, separate with the covering around it. Like classroom must be divided with the ages and the knowledge of the students. So that compartmentalization make the function more systematic. In the same way, in the complex level in eukaryotes, the every structure which is still inside the cell, they are having compartments due to the membrane around it, and they are performing their function within that limits, right? So that membrane bound structures are present in case of the eukaryotic but absent in case of the prokaryotic. So which two differences we discussed? Number one, the well developed nucleus is present in eukaryotes and absent in case of the prokaryotes. Membrane bound cell organelle is present in eukaryotes but absent in case of the prokaryotes. So which is a primitive type? Prokaryotes. As I said you, the pro means for primitive, karyon for nucleus. Then what do you mean by the eukaryote? Eu means true, karyon means nucleus. Means it is having the true nucleus and membrane bound structures are there. Is that clear to everyone? So what is there? There are the two types of cell, prokaryotic cell and the eukaryotic cell, right? In the eukaryotic cell, mainly we will discuss in this chapter regarding the plant cell and the animal cell, right? Under the prokaryotic cell, who will come? All type of bacteria will come here, right? So this bacteria, these are what thing? Prokaryote. As an example, even just now I said you mycoplasma is there, no? even it comes under the prokaryotic. Many more things you will learn when you will exemplify it with much more higher levels, right? Just now you should know that bacteria is a prokaryote or a eukaryote. It is a prokaryote. If I am saying the plant cell, animal cell, which type of cells these are? Eukaryotic cell. Or can I say plant itself are eukaryote and animals are itself as a eukaryote. Whereas the bacteria and the mycoplasma, like examples are what? Prokaryotic cell, or you can call it as prokaryotes. Is that clear to everyone? Okay. So now next what we have to go with? Now please have a look on this structure in front of you, that is a plant cell. The plant cell, you know that it is green, photosynthetic in most of the autotrophic organism. What word I used here? Autotrophic. Auto means what thing? Self. Troph means food. Right? Those organisms which can make their own food, right? They are mostly the plants, right? But still we will discuss the various type of lower organism which can make their food. So mainly maybe you are aware of the plant, that's why we are discussing about plant cell of the higher plants. Like all the plants around you, like angiosperms are there, gymnosperms are there, that all we will take in a higher plant. Now what is gymnosperm, angiosperms? We will discuss in the plant kingdom according to the classification. Fine. So what the features of that, we will gradually come into all the topics one by one, right? But keep in your mind, not in much detail, only superficially we are touching all the points one after the another, right? Now what we have discussed till now, the introduction of cell, what is a cell? Then we have discussed the scientist name Robert Hooke. Can you say me which type of cell Robert Hooke? Robert Hooke has discovered which type of cells? Anyone? Which type of cells Robert Hooke discovered first? Dead cells. Everyone, I need your attention, your response here. Cock is an organ, what he had taken, right? Whereas the cell are the dead cell over there, right? Cock cells, or you can say cock cells, right? But not the living cell. He discovered first the dead cell. And let me to say you one more thing. Robert Hooke was the person who had introduced the term cell. Robert Hooke was the person who introduced the term cell. Actually he given the term cellular. From the cellular we get the term cell. Cell means what thing? Little room. When Robert Hooke cut its section, he found that cells are like little rooms, and he given the term as a cellular first of all, which slowly change into which term? Cell, right? So that thing you should know about that person. Then after this, right, what is this cell? Animal cell. Just now I said you the plant cell and animal cell, this category will come under the prokaryote or eukaryote? Eukaryote, right? Okay, now we have to discuss the main features of the cells. First of all, we will discuss about prokaryote, then gradually we will discuss about eukaryote. Let me to take the more description first of all, prokaryotic cell, then I will take the description of the eukaryotic with the difference between plant cell and animal cell, right? So let's see for prokaryotic cell now. In a prokaryotic cell, innermost what is there? This you call it as cell membrane. This you call it as a cell membrane. The layer which is innermost. Outer to the cell membrane, what is present over there? Cell wall. Here is the presence of cell wall. Still outermost what is present? There is a loose layer which you call it as slimy layer, glycocalyx, right? Small term we are introducing gradually, so that you should aware of something when you are coming here. So here you are having the outermost, which you call it as glycocalyx, right? Just focus on the terms what I am describing you. Don't go in much detail of the topic. I repeat, which is the outermost? Glycocalyx. And which is innermost? Innermost is what thing? Cell membrane. And in between the outer and the innermost, what is there? Cell wall. Now it is there in your mind till now that plant cell is having cell wall. But can you see here, this prokaryotic cell can even have the cell wall, where I am discussing typical bacterial cell. Let me to say you what do you mean by typical bacterial cell. Typical bacterial cell means we are supposing it is having all the characters of different bacteria. Some bacteria may be having not one feature, may have other feature, right? So we are assuming all the feature of all the bacteria in one cell. That you call it as typical bacterial cell, which is a prokaryotic. So we started discussing the cell structure of the prokaryotic cell, right? Glycocalyx, simply you know, it is the outermost layer which can be loose or it can be like a hard. When it is loose, you call it as a slime layer, and when it is hard, you call it as a capsule. When the bacteria need to be hard from outside, when it is going in the body of the any of the you can say host like plants, animals, where they have to put the infection. So obviously, like if it is coming in our body, our immune system is going to fight against it. To resist against that type of immune, what we are secreting against it, they are making themselves hard. But if it is outside, what I am discussing here, let me to repeat, we are discussing about the bacteria. I repeat my point, bacteria when enters, right, it is not viruses, it is the bacteria. When it enters in the body of the host, either of plant, animals, when they are going to attack, our immune system will fight. To resist against the immune system, they become hard. Then that glycocalyx, you call it as a capsule. Otherwise, if it is free in the environment, they are having everything sufficient, then it become a loose slimy layer. That you call it as what thing? Slime layer, right? So which is the outermost layer here right now? That is a glycocalyx. Inner is the cell wall and innermost is what thing? Cell membrane. Now this cell wall is enclosing the cell membrane, and this cell membrane is making inside the loop like this, invagination towards inner side. That you call it as what thing? Mesosome. I hope so you know a little bit about mitochondria and eukaryotic cell. Mesosome is behaving as a mitochondria for prokaryotic cell. Otherwise, it is having much more other functions to perform other than the respiration like in mitochondria. Let me to say in detail, the mesosome is like, or you can say it is equivalent to, it is functionally like mitochondria of eukaryote, right? What function? Like for respiration. Otherwise, it is having other functions as well. I will discuss all the functions with the some topics which is very important regarding prokaryotic cell. So what is the invagination of the cell membrane? That is the mesosome, and it is having much more other function. But which function is similar to mitochondria? Respiration. Are you getting that? Yes, you call it as powerhouse of the cell. But that mitochondria is present in eukaryotic cell because it is membrane bound. Just now I said you membrane bound are present in case of the eukaryotic cell. Are you getting that? So mesosomes, what other function we will discuss later, but right now you should know about this structure. You call it as mesosome. It is not the invagination of cell wall, it is not the invagination of glycocalyx, it is the invagination of cell membrane, right? Okay, now coming to this point, who is controlling everything inside it. As nucleus is not well defined, it is having the genetic material like this, which you call it as a DNA. What is DNA? This is a nucleic acid which is controlling the features, like what we appear. This is a prokaryote, but I am talking about we people, which we are eukaryotic. Even in our cell, nucleus is controlling our activities. Why? Because nucleus is having this genetic material DNA, right? What is the full form of DNA? Do you know already? Anyone can say the full form of DNA? Anyone? I am waiting for your response. Genetic material, very nice, it is a genetic material. It is the dinucleic acid, deoxyribonucleic acid, very nice. So here that you call it as deoxyribonucleic acid. This is acting as a genetic material. Are you getting that? So DNA is a genetic material which is double stranded. Da stands for the double stranded, and it is not surrounded by, what I mean to say here, it is not, cross I mean to say not, it is not surrounded by nuclear membrane, right? It is not well developed. Now coming to the next structure, apart from this DNA which is present in a region, region means area, that area you call it as nucleoid, that area you call it as nucleoid. Just now I spoke to you, what do you mean by the word oid? I discussed that is amoeboid. Oid stands for what thing? Just now I spoke to you. Hurry up, give the response. Oid, OID, oid. As a suffix if we use, oid means what thing? Like. We discussed, very nice. So we discussed already, like where amoeboid, shape of what thing? White blood cells. There we discussed, no? White blood cells are amoeboid, amoeba like. So here they appear means it seems to be nucleus like, but actually there is no proper nucleus. That area, very nice, everyone is responding now, that's good, okay. So here nucleoid is a region where this DNA is present and controlling the activities of its cell. Now apart from this DNA, more DNA is there which is circular in shape. This you call it as extra chromosomal DNA, right? What is a chromosomal DNA? This one you call it as chromosomal DNA. Extra means apart from this add-on, a little more is there which is separate, that you call it as extra chromosomal DNA. And what is the other name of that? Plasmid. What is the other name of it? Plasmid. Even this is controlling some characters, not all but some special characters, even it has capacity to control, but major control is in a hand of the nucleoid. Are you getting that? Now obviously what is present around all these special things inside, that is all cytoplasm, right, which is the main arena of, this is the arena of metabolic activities, main arena of metabolic activities. What do you mean by arena? Arena means place where the major things are happening. So metabolic activities means either things are breaking to create simpler thing, or it is joining to form the bigger one, right? So I am talking about catabolism, anabolism, which we'll discuss later. Simply the chemical reactions which are happening in a living organism, their main place is what? Cytoplasm, right? So that cytoplasm is an arena of metabolic activities. Fine? Okay. No, DNA is not ribonucleic acid. DNA is deoxyribonucleic acid, RNA is the ribonucleic acid, which is another type of nucleic. But the genetic material in all living organisms, prokaryotes or eukaryote, it will be DNA only, right? RNA is even present here, but when it will be going to form, we will discuss in higher topics, right? Okay, not area, here I am talking about arena. Arena means place, right? Okay, now coming to the next part, we discussed about the cytoplasm. After this, they are having some structures which you call it as what thing? Ribosomes. Ribosomes, that you call it as a protein factories. Protein factories of the cells. Protein factories of the cell. Proteins are giving the shape, structure, they are making the enzymes which are helping in the reactions in our body. That ribosomes are the factories of such a good type of the macro thing, bigger thing, that you call it as proteins. So which type of ribosomes are there? 70S. Simply just learn here 70S. Detail why you call it as 70S, we will much more discuss in our higher more topics within the same heading, cell the unit of life, right? So which type of ribosomes are there? 70S is a main feature of prokaryotic cell. So this ribosome obviously is membraneless. There is no membrane around any structure here in prokaryotic cell. So whatever we are discussing, that is all without the membranes, right? Yes, this is a site of protein synthesis. Ribosome will synthesize the protein. Very nice. And what type over here? 70S is there. Clear? Okay. Now that we have discussed the things which are inside. Some projections are there outside even. Inside invagination is what? Mesosomes. Even something is projecting outside. What are that? Maybe it can be the tail-like structure which you call it as what thing? Flagella. What do you call it as? Flagella. Like this. Whip-like structure which help in the movement, right? And even you can find some of the structure. Let me to draw on the next more diagram. The outer extending thing. What is inside and invagination I discussed with you. Now what is projecting out? I will say you through next diagram. But I hope so this all is clear to everyone. Please put your response here. Is that clear to everyone? Hurry up, give your response here. Okay, fine. So here we are discussing about these structures. I hope so the things are clear to you. Let me to give on the next slide much more, a little bit expansion of this term mesosome. I said you it perform other function. What are the other function? It is even helping in cell division. It is even helping in absorption. Even it is helping in secretion. Right? And even it is increasing the surface area. I said you even it is helping in respiration. So on this function behalf, this mesosome is like the mitochondria of eukaryotic cells. It is equivalent to that thing functionally, right? Not structurally. Now coming to the next part, let me to take you on the previous diagram. Let me to expand this thing even which you call it as ribosomes. A small description of that. If I am talking about RNA, the full form of RNA is ribonucleic acid. This is of 3 types, but one type is like that which you call it as mRNA. mRNA stands for messenger RNA. Right? 3 types we will discuss later. I am taking only one type here. What is that? mRNA. M stands for here messenger RNA. Now this messenger RNA is sent by DNA, and around that what is started accumulating? 70S ribosome. These all are which ribosome? 70S ribosomes. Can I say the 70S ribosomes are many? But messenger RNA is only one molecule or one strand of that will be there. This complete structure you call it as what thing? Polyribosome. What do you call it as? Polyribosome. So what polyribosome? Poly means many. Many ribosomes are present on single molecule of mRNA. When such structure is ready, then obviously there will be something formation. What is that? Protein formation. So for the protein formation, messages are coming from DNA with the help of mRNA, and that mRNA need the ribosomes on it and help in the formation of proteins. Are you getting that? Structure and transcription. Yeah, we will discuss all these terms, transcription, translation later on. Adsorption we are not discussing, we are discussing here absorption. Absorption means taking things from outside. So if these are going to be more, the mesosome infoldings, they will take up more things from outside, right? Prokaryotic cell can show cellular respiration, that's true, but aerobic and anaerobic we will discuss later in our higher classes. What do you mean by 70S? That is a big concept behind that, we will discuss later on. Meanwhile, I simply can say you, if you centrifuge the thing at very high speed, can I say heavier will settle down faster, right? And the lighter will settle down, going to be taking more time. I repeat, lighter will take more time to settle as compared to heavier. So heavier will come at the base and lighter will come at the top. So that values is determined by S, right? And that you call it as Swedberg's constant. We will discuss later on about this. It will be out of now the basic things, right? Don't take tension this much right now. What I am saying, take it only that basic thing so that you can enter here with a full confidence. We have still to go with the full chapter again with more details, right? Okay. Okay, all the function is not performed like the mitochondria. One student is asking, can mesosome do all the function like mitochondria? It cannot be, because we will discuss in respiration what is the complexity in its structure of mitochondria. Definitely it will going to be much more complex in the eukaryotic in mitochondria as compared to mesosome. But the main motive behind that to provide energy for function, that is up to mark done by the mesosome. Fine. Nucleoid function, it is containing the DNA, it will control the function of the full cell like the nucleus in eukaryote, right? mRNA full form. mRNA full form means messenger RNA. What is the full form of RNA? Ribonucleic acid. Have you got this point now? Please put the thumbs up, those who are asking the question and it is clear, right? Okay, powerhouse of the cell is mitochondria. Fine. Okay, now coming back to the topic. So this is about the basic inside and invagination. Now what is outside? For outside, this is a cell membrane, right? I am taking this as a cell wall, and outside what is there? Glycocalyx layer. Fine. If we are getting whip-like structure like this, that you call it as what thing? Flagella. Singular you call it as flagellum. Flagellum is singular. Even you call it as what thing? Flagella as a plural. Fine. Even it is having, this is for what thing? This is for locomotion. Whereas other, these types of structure which you call it as a fimbriae or pili, these are different terms. These are different terms, right? They are helping in what thing? Attachment. These two are helping in attachment. Means when bacteria attack on a host body, it is going to create the infection there. So it will hold on that host with the help of the fimbriae, right? but sometimes bacteria is holding with the bacteria, that is by the pili. So pili is holding the bacteria with bacteria, or you can say bacterium. Bacterium is singular, and this is holding the bacteria or the bacterium with what thing? Host, or you can say the host cell, or with any other substrate. Substrate means any other ground. Fine. So is that clear to everyone? Fine. So these are the extensions which are going outside. So this is a basic thing, at least you should know what is there in a prokaryote. The nucleoid is there, plasmid is there. Plasmid is even performing very important function. Like bacteria, when it is entering in our body, we are eating antibiotics to kill the bacteria. But bacteria can even save itself, right, by fighting against that antibiotic. That gene, that control is in hand of the plasmid. Many other such controls we will discuss later. Just simply to satisfy with you the function of the plasmid I spoke. So can I say this plasmid is made up of what thing? This plasmid. Again it is made up of what thing? Double-stranded DNA, and controlling further important functions of the prokaryotic cell. Right? So this is all about the prokaryotic basic structure. Now coming to the eukaryotic cells, as per I already mentioned you, this is a plant cell, right? And this is what thing? Animal cell. Just have a look on this diagram. But what is the labeling of that? What is part? We will gradually come into this topic, right? Now let's see the differences now. First of all, what are the differences between plant cell and animal cell? First of all, can you see the basis, the difference in the size? Usually the plant cell is larger as compared to animal cell. But both are eukaryotic cell. We entered into the discussion of the topic of the eukaryotic cell. Fine? Okay. Now coming to the next, cell wall. Cell wall is present in case of the plant cell. It is absent in case of the animal cell. Plastids. Plastid means what thing? Which help in the photosynthesis. For example, here let me to take the chloroplast, that are present in the animal cell, absent in case of animal cell. I repeat, it is present in the plant cell and it is absent in the animal cell, right? Coming to vacuoles. Vacuoles will be in both, but it will be very large and it is going to be single in the plant cell, whereas it will be many and small and sometimes temporary form in case of animal cell. Right? Big and large, one only will be in case of the plant cell. In the mature plant cell, what it can be many, small and temporarily formed structure in case of animal cell. Right? Now Golgi bodies, we will discuss what is that and its function in the big description. It is going to be very simpler in case of the plant cell. There I will discuss in the description how it becomes the simpler, but complex it will be in case of animal cell. Right? Now centrosome and centrioles. This simply right now you should know they help in cell division. They help in cell division. So it will be present in animal cell but absent mostly in case of the plant cell, because lower plants can have this centriole. Now what is this? All terms we will gradually enter into all these things, right? So at least keep in your mind that plant cell, they will have plastid, they will have cell wall, but that cell wall and plastids are absent in animal cell, right? What thing is more present in number here? Vacuoles, and even they have in all the cells centrosome and the centriole in the animal cell. And in plant cell, these generally centrosome and centrioles are absent, right? Now coming to the next part, when we are taking the cell into the study, can I say even the plant cell, the outermost layer is what thing? Plant cell if I am taking, can I say the outermost layer is cell wall? And what is inner to cell wall? Cell membrane. It is having cell membrane, right? But in animal cell, it will start with the directly with the cell membrane. Let me to draw it is more in oval in shape, right? Usually plant cells are angular in shape, they have some angles, but animal cells are usually oval and elongate, usually it is like that. And this is directly having the plasma membrane, which you even call it as cell membrane, right? Here even you can call it as a plasma membrane or cell membrane, right? So can I say animal cell is starting from the outermost layer plasma membrane, whereas plant cell is starting from the cell wall. Now what is the basic difference between the cell wall and the cell membrane? Let's see that differences. If I am taking for cell wall, can I say it is present in plants? If it is present in the plants, then it is made up of which thing? It is made up of cellulose. Even it can be present in the fungus, where it is made up of chitin. What is chitin? We will discuss in the biomolecules chapter. What is cellulose? Again we will discuss in the biomolecules. But can you see cell wall is there in these, but the composition is different, right? And even it can be in some protist, it can be there. Just now we discussed, even it is present in the bacteria. It is usually made up of what thing there? If I am discussing the eubacteria, in classification you will discuss different type of bacteria, eubacteria, under eubacteria, cyanobacteria, even mycoplasma, actinomycetes, right? So that all categories comes under monarens, right? I repeat, these all categories are coming under Monera kingdom, right? Actually R.H. Whitaker has classified all the living organism into 5 kingdoms: Monera, then after Protista, Fungi, Plantae and Animalia. So Monarens containing maximum the bacteria there, right? And bacteria category can be eubacteria, right? Even the monarens contain the mycoplasma. Just now I said to you, the smallest cell is of what thing? Mycoplasma, which even you call it as PPLO. So here I repeat, the bacteria which is a eubacteria, which is in which kingdom? Monera. Their cell wall is made up of peptidoglycan. Here it is made up of peptidoglycan. Fine. Now I have said you the presence of cell wall. But if I am saying cell membrane is present in all living cells, right? If all living cells, it is present, whether it is a prokaryote or eukaryote, they have the living cells, means they have the cell membrane in the living cells. Fine. Okay. Now coming to the next part, what is a composition? Means how it is made up of, which material it is made up of. In case of cell wall, the composition I already mentioned. Here can I say it is made up of lipoproteins. Lipoproteins means it is made up of lipids and proteins, right? And the cell membrane is living, whereas the cell wall is dead. Are you getting? What is the function of the cell wall? Function of the cell wall is to provide rigidity, protection and shape to the cells. Whereas this thing helps the selective permeability of the cells. Now please focus on these new terms, maybe these terms are new to you. Rigidity, obviously stiffness to the plant is provided by what thing? By the cell wall. Whereas selective permeability, to select, everything can cross through the cell wall, but they cannot cross the cell membrane. Cell membrane will allow that thing only to enter which is required, and from inside of the cell to outside, which is extra, which is not required, or it is going to be the waste, that will make them out. So who is selecting that thing? That is the cell membrane, which you even call it as plasma membrane. So this is like a function of both. Are you getting that? So can I say these are what thing? These are the feature of cell wall and the cell membrane. So chemical composition can vary depending upon which organism is having cell wall. But cell membrane is present with the same composition of lipids and protein in all living organism, whether it is prokaryotes or eukaryotes. Can I say common thing between prokaryote and eukaryote is that this cell membrane is completely same. Somewhere one or two small differences are there that we will discuss later, right? So this is a basic difference. Let me to show you about cell membrane. It will be having this type of structure, right? So what is there? Can you see the ball-like structures here? What is this ball-like structure? These are the head part of the lipid. This lipid is having two parts, head and the tail, right? So this is one molecule of lipid. Like that, many molecules are going to be close to each other, but it is forming two layers. Two layers of lipids are there, and you call it as lipid bilayer, right? And this lipid bilayer is continuous like this, but in between it is interrupted by what thing? By the proteins. If the proteins are only at the upper side, that you call it as peripheral. Integral means which is embedded like this. Two types of proteins are there. One is peripheral, which is toward outside, and this is integral means intrinsic. Intrinsic means which is inside, embedded in the lipid bilayer, right? So main composition is what? Lipids and the protein. But somewhere you will find the sugars like this, chains of the sugars will be there, but its percentage is very less. The main composition is of lipid and the sugars. Main composition is of, let me to repeat, correct myself, main composition is of lipid and the proteins. Sugars are somewhat there, right? But not in a main composition. Main composition is lipid and proteins. Are you getting that? Okay, so detailed structure of membrane is studied in which year? 1950s, right? This model, what is in front of you, that was given by Singer and Nicholson, right? This model, or you can say presentation of the cell membrane, is given by whom? Singer and Nicholson. What is lipid? Lipid, we will discuss, is a micromolecule which is having the hydrophilic, hydrophobic tails. If I speak more, maybe you will get confused. Simply you should know this is one of the biomolecule which is present in our cell, and it's making the main composition even in the membrane, right? So we will discuss its structure, its detail, everything much more in biomolecules chapter, right? So this is a basic outline for the structure of cell membrane. Now coming to inside, what is inside the cell of eukaryote? Please see here, inside the cell, what is present? Inside the cell, let's start, the organelles are present. Which thing is present? Organelles. But we are discussing the eukaryotic cell. So which organelles are present? Membrane bound. Membrane bound cell organelles are present, right? But we are taking first of all endomembrane system. Endomembrane system includes what thing? Endomembrane system includes endoplasmic reticulum, which you short call it as ER. Fine. So what you call it as ER? Golgi body is second. Third is what thing? Lysosome. Four is what thing? Vacuole. Four structures are coming under the endomembrane system. So can you see where the endoplasmic reticulum? This part we have shown you the nucleus, right? Suppose this is a complete cell, and in the cell this is a part of nucleus we have shown you, right? Let me to take like this is a complete cell and there is a nucleus. The nucleus part we have projected, right? So nucleus, can you see, is immediately surrounded by endoplasmic reticulum. We will discuss endoplasmic reticulum are of two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. I will go in detail, but at least you see the position where it is present. I repeat, we have taken a fraction of the cell here. If I am taking this as a complete cell and this is the nucleus, can I say immediately what is surrounding it? That is the endoplasmic reticulum. And what is this here? Golgi bodies. And this, what is present? Lysosome. And what are further? Vacuole. Actually they are interconverting into each other when the processing happen. These both are the vesicles. These both are the vesicles, like small pouches. And what is their function? How it is formed? That we will discuss in its detail, right? So just focus right now what is endomembrane system, which constitute of these four organelles. Why we are including this into endomembrane system? Because they are coordinating with each other functionally. Functionally they are coordinating with each other. Coordinate function means, let me to give you one example here. The function of RER, which you call it as rough endoplasmic reticulum, is what? It is having on it, why you call it as rough? Because it is having on it the ribosomes. What they are having? Ribosomes are there. And you know that ribosomes are the protein factories of the cell. But here the ribosomes are ATS. Which ribosomes are there? ATS ribosomes are there. Keep in your mind here, ATS ribosomes are present in the cytoplasm as well, and even they are present on the RER. What is the full form of RER? Rough endoplasmic reticulum, right? So what is happening? They will make a protein. What is the responsibility of ribosome? To make the protein. So RER is forming protein, but it is sending with the help of small vesicles. This RER sending by small vesicles that protein into the Golgi bodies. The Golgi body is going to process that thing, is going to modify little bit, store it, then dispatch it again within the cell or outside the cell. Are you getting? Not only protein, but even one more thing which we will discuss, which is formed by SCR. Have you got this idea? Right? So they are coordinating with each other. We will discuss gradually that what is the function of lysosome. You are asking the function of lysosome, we will discuss. But right now I am giving you the meaning of endomembrane system, how they are coordinating with each other. Where the first we will take endoplasmic reticulum, then Golgi bodies, then lysosome, then the vacuole. But can you see the position right now? Especially RER is surrounding the nucleus. SCR will be slightly away from that, right? Let's see the description now. If I am discussing with you endoplasmic reticulum, right? It is made up of mainly this cisternae like this. They are linked with each other like this, right? And having some tubules in their surroundings. Like this is a basic structure of endoplasmic reticulum, right? And they are giving out the small vesicles. So what thing I have drawn? These are what thing? Let me to use the other color for this. Cisternae, and this you call it as tubules, and this you call it as vesicles. This is the basic structure of the endoplasmic reticulum, right? But how many types of this are there? Two types are there. What are the two types? That will be the RER. Just now I said you what is RER? Rough endoplasmic reticulum. And what SCR stand for? That you call it as smooth endoplasmic reticulum. I already said you why it is rough in surface. Why it appear rough in surface? Because they are having ATS ribosomes. So what is the function of this rough endoplasmic reticulum? They are forming the proteins. Fine. Now SCR, smooth endoplasmic reticulum, they do not have this ATS ribosome on it. Then what is the function of this? That they are helping in the formation of lipids, right? So lipid formation is helped by the SCR and the protein formation is by RER. Now all the thing which is going to be formed, it has to enter into next organelle, which you call it as Golgi bodies. This is after the name of scientist Camillo Golgi. You call it as Golgi apparatus, or even you call it as Golgi bodies, right? This is named after the name of scientist Camillo Golgi. That is the name of scientist, right? And what it is doing? It is going to help in modification of material which is formed by what thing? Endoplasmic reticulum. Even it is going to store that, even it is going to dispatch that. Dispatch will happen in the form of vesicles, right? Now what is the structure of the Golgi bodies? Even it is made up of this cisternae, like this, one is piled upon the another and it is linked with each other. Many cisternae piled upon on each other. Whatever is coming from ER need to be processed. Now please listen this point here, this is very important what now I am going to say it. Even this is having cisternae, even they can have the tubules, right? Even they can have the vesicles, right? Endoplasmic reticulum mostly after forming the thing is sending to the Golgi bodies for its modification and finally its dispatch. So here this structure, can you see this is a convex side of it and this is the concave side. The convex side is towards the nucleus. It faces towards the nucleus, and this faces towards the membrane. It is facing towards the membrane. Like now I am facing towards you, right? So suppose this is a cis, or you can say the convex. Cis and trans is a much more level. Simply just now you know this is the convex side which is facing towards the nucleus. How it will be? Let me to take like this. Please see here once again. Suppose this is the cell and here is what thing? The nucleus is present. Then this Golgi apparatus having this convex side facing towards, can you see this convex side bulging is toward the nucleus, because it is receiving the material which is coming from rough endoplasmic reticulum. It is surrounded by what thing? Rough endoplasmic reticulum, right? So whatever is formed by rough endoplasmic reticulum has to enter here. So entering of the material is happening towards the convex side. After the modification it is coming from which side? Concave side. Fine. Have you understood this polarity? Polarity means the facing of the Golgi bodies, where it will be convex side, bulging side is toward the nucleus and this concave side is facing towards the cell membrane, right? I am not saying it is on the cell membrane, I am saying just facing towards the cell membrane. Fine. Okay. Then after this, let me to go with the next point. We discussed what is the next cell organelle there. First is endoplasmic reticulum in the endomembranous system. Second we discussed Golgi bodies. Now let's see the lysosome. Lysosome, even you call it as a suicidal bags of the cell, right? Suicidal bags of the cell means if cell is not working within a tissue, it has to be disposed of. Means they are containing, lysosomes are containing some enzymes. What do you mean by the enzymes? which help us to fast the reaction. Like in chemistry, maybe you have learned about catalyst. So that catalyst in human body does not same, but it will be slightly different. Slightly, I mean to say, based on its structure it will be much different, but functional it will be same to speed up the reaction. So that enzyme help to dispose of the thing or making of the thing very easily, but here we are talking about disposing of. If the cell is not working, it will be disposed of by which organelle? Lysosome, right? So if cell is not working, suppose cell is okay, fine, only organelles become non-functional. So that will be digested, that part only will be digested again by the lysosomes, right? But it is made up of enzyme, and enzymes are made up of what thing? Proteins. Now who is forming the protein? Please give me the answer. Who is forming the protein? Hurry up. Who is forming the proteins? Hurry up, please give me the answer very fast. Ribosomes. But ribosomes are present right now on which endomembrane system? See, our main topic is about what thing? Endomembrane system. So which endomembrane system have ribosomes on it? I want that answer. Which endomembrane system is having ribosome? Rough endoplasmic reticulum. Very nice tree. So here will be the rough endoplasmic reticulum, which is having the ribosome and helps in the formation of protein. Can I say that proteins are formed by endoplasmic reticulum, which is rough endoplasmic reticulum, but send to the Golgi bodies for if any change is required, then in the vesicle it is going to be released. That vesicles you call it as lysosomes, and lysosomes work in acidic medium. They are having the acidic medium for the functioning of such enzymes, right? So this much you should know about that thing. Then vacuole. What is vacuole? See here, this is even all the structure. One more thing let me to say you here. If I am taking all the endomembrane system, what are that? Endoplasmic, Golgi bodies, lysosome and vacuole. Here all are single membrane bound. All are single membrane bound structures. Are you getting that? But now I am discussing right now with the lysosomes which have been formed from RER. Getting that? RER is even the single membrane bound. But RER is present on that what thing? Ribosomes. Which ribosome? ATS. But that ribosomes are membraneless. That is membraneless. Where it is present? On RER. So it helps in the formation of protein. Some of the protein which is required here, right? Among so many proteins which is there in a cell, even this is formed by RER. Now coming to the vacuole, even this is single membrane bound. Now this vacuole will be single and large in case of plants, but it will be more in number but smaller in size in case of animals. Can I say animal cell? Here I can say the plant cell. Fine? Okay. Nice. You are giving me the answer. That's fine. So here the lysosome and vacuole we have discussed. Now what is the function of vacuole? You know the function of kidney in our body. So it is going to be acting as osmoregulatory organ which will give out all the non-essential ionic forms or whatever substances are not required, even water, to remove out. Same here is a function of the vacuole in the cells which will act as osmoregulatory part of this cell. Are you getting? So different types we will discuss. Contractile vacuole is there, food vacuole is there, but overall if I give you the one line function, the main is what thing? Osmoregulation. and especially like if I take amoeba, amoeba usually is in water, excess of water can enter. So that excess will enter into vacuole and it will expel out from the cell. There we call it as contractile vacuole. Now what word I used here? Contractile, contractile vacuole, right? Now food vacuole, second vacuole, what type? Food vacuole, many other types, but mainly you should know these two at least. Food vacuole is what? Like when amoeba is capturing the food, it is coming in a form of vesicle, and that vesicular form you call it as what thing? Food vacuole. Are you getting that? Okay. Golgi bodies apparatus function, I said you, what is formed by endoplasmic reticulum will enter into the Golgi bodies with the help of vesicle. There it is going to be modified, stored and dispatched finally by Golgi bodies, right? And more if you want to know, we discussed in the beginning that Golgi bodies are very simple in case of plant cell. In plant cell, that Golgi bodies you call it as dictyosomes. Dictyosomes term you use in Golgi bodies in case of plants. But as such, I am discussing about right now about the Golgi bodies. That is known as dictyosomes in case of plant cell. But in animal cell, we name it as Golgi bodies. What is dictyosome? Like membrane after membrane we are drawing it now. If these are separate from each other, no piling, no clustering, if it is not there, that you call it as dictyosome, but function will be same. But clustering together like this, pile upon each other, that is in case of animals, and that you call it as Golgi bodies or Golgi apparatus. I hope so it is clear now. Okay, fine. Now coming to the next point, we were on here. Just now we completed the endomembranous system. First we discussed endoplasmic reticulum. Second we discussed about Golgi bodies. Then we discussed about lysosome. Then we discussed about the vacuole. Now apart from this, what more are there? Can I say apart from this, they are having the powerhouse of the cell. Now let me to remind you, we have completed the endomembrane system. Now what else apart from endomembrane system? That is a double membrane bound structure which you call it as mitochondria. Mitochondria is double membrane bound structure having outer membrane, right, and even inner membrane. Inner membrane is having such foldings like this, right, inner membrane, right, like this. And these folding you call it as what? Crista, right, plural you call it as what thing? Crista, right. That is there in case of the mitochondria. It is double membrane bound structure. Very important question. This is double membrane bound. Chloroplast is even double membrane bound, right. Now here the outer and inner membrane divide itself into the two aqueous compartment, outer and inner compartment. Maybe this picture which is given in your NCERT book is not going to be much clear in this concept. That's why I am drawing here, right. This part, can I say this is outer compartment, right, and this you call it as what thing? Inner compartment. If I show you from this side, this part is what thing? Inner compartment, and this one between two layers, this is what? Outer compartment. Fine. So right now it is double membrane. Dividing the mitochondria is two compartments, outer compartment and inner compartment. Inner compartment you call it as matrix. What do you call it as? Matrix, or even you call it as mitochondrial matrix. And what is the name important given to the mitochondria? You can call it as a powerhouse of the cell. It is known as powerhouse of the cell. Why you call it as powerhouse of the cell? Because it is producing ATP. What is ATP? That is the energy currency of the cell. Like when we are eating the food, right, it is burning, it gives us energy, but instantly if we are not using that energy, it is stored in the form of ATP. What is the full form of ATP? Adenosine triphosphate. Adenosine triphosphate. Is that clear? See, mitochondria you call it as powerhouse of the cell, whereas ATP you call it as energy currency of the cell, like currency you spend when only you required. In the same way, ATP is going to be spent only when it is required. One more interesting thing, the mitochondria, they are having inside 70s ribosomes. Even they can have small amount of DNA as well, which is double stranded. See, the ribosome 70s can present inside mitochondria, but 80s is present in case of the cytoplasm and the rough endoplasmic reticulum, right? Due to the presence of this DNA, you call it as semi-autonomous cell organelle. Are you getting that? You call it as semi-autonomous cell organelle. Semi means, please focus here, semi means what thing? Half. Auto means self. Due to its DNA, of its own DNA, they can do half function by its own without the control of nucleus. Are you getting that? So these are the features of mitochondria, at least you should know about this, right? Now coming to the next, for the chloroplast. Chloroplast is present in case of plant cells, not in the animal cell. So what we have discussed till now up to mitochondria, it is present both in case of the plant cell as well as animal cell. But cell wall we discussed, that is mainly in the plant cell. Why mainly? Because other fungal members, even protistans, have cell wall. I am talking in that aspect. Fine. Now see here, this chloroplast, the material inside that you call it as stroma. Actually this is a type of plastid. We will discuss the plastids are of three types, that is the leucoplast, we will discuss about chromoplast, we will discuss about the chloroplast. Right now we are discussing about the one of the plastid. I am talking about plastid, not plasmid. Plasmid we discussed as a DNA in bacteria, the circular one I have drawn, extra chromosomal DNA, that is plasmid. Now I am taking what thing? Plastid. So chloroplast, inside it is having which thing? Stroma, right? In the stroma they can have carbohydrate and protein. Can you see, even this is having DNA which is double stranded and ribosome as well, same like mitochondria. That's why you call them as what thing? Semi-autonomous cell organelle. Semi-autonomous cell organelle. Because they can perform half function of their own, like mitochondria, right? But keep in your mind, they have 70s ribosomes. Clear? Okay. Now what is still inside that? Now what is the function of leucoplast and chromoplast? Can I say they help in storage only? Chromoplast is providing color other than green, but main photosynthesis is done by what? Chloroplast, and it is green in color, being main pigment are present in chloroplast. So we are discussing that by chloroplast in much more detail, right? Okay, now coming to inside, what is present? Inside, can you see they are having like coins like thing coiling upon each other, not coiling, I must say piling upon each other. The one sac like structure you call it as thylakoids. Now these thylakoids make this bundle that pile upon each other, that you call it as one as a granum, single as a granum, plural as a grana. Many grana can be there, right? And what is there in one thylakoid? If I take one thylakoid, the space between that you call it as lumen. The space between that you call it as lumen. Whereas the space in the chloroplast or the plastid, that you call it as what thing? Stroma. What you call it as? Stroma. And the space inside or material inside the mitochondria, you call it as mitochondrial matrix. Are you getting that? Okay. So can I say they even having in their stroma, they have DNA, If DNA is there, definitely RNA is there, even proteins will form, even they are having 70S ribosomes. But add on, they are having starch. This starch is absent in the mitochondrial matrix because it is not showing photosynthesis. Please keep in your mind, this is very important. Rest of things are same in mitochondrial matrix and stroma, but starch is the main component of the chloroplast, right? Okay. Now coming to the next part. After this, types of ribosome we already discussed, where the 70S are present, found in prokaryotes and eukaryotes. Eukaryotes we are just now discussed in case of the chloroplast and in case of mitochondria, right? Now 70S actually made up of two components, 50S and 30S. Just simply learn that. Don't go in much depth. And 80S, where it is present? 80S is mainly a feature of eukaryotes. Eukaryotes, where it is present? It is present on RER and in cytoplasm. And already I said you, what is the function of ribosome? This you call it as protein factories of the cells. And this is membraneless structures, but present in both prokaryotes as well as eukaryotes, right? So it is having subunit 60S and 40S. By joining that, when they join, then protein synthesis will start, either in eukaryotes or in prokaryotes. Is that clear? Okay. Now coming to next part, something more is there, cytoskeleton. Think like a room is there, it is covered by the webs. In the same way here, in the cell, like the webs, some proteinaceous interconnected structures are there, which is very thin, that you call it as what thing? Cytoskeletal system, which is network, elaborate, means throughout the cell, made up of protein, and it formed the framework inside the cell. And this feature is of only eukaryotic cell. It cannot present in bacteria, means it cannot present in the prokaryotes. Prokaryotes, they are not present. And what are that? Three: microtubules, microfilament and intermediate filaments. Only you should know right now cytoskeleton. Cytoskeleton means like throughout the cell, what three structures are there? These three structures are there. Now what is the function of cytoskeleton? The function of cytoskeleton will be this. You can learn it by 3 M's. 3 M's are there. What are that 3 M? One is the mechanical support, motility and maintenance of the shape of the cell. Together this is the function of cytoskeleton, right? Now last not the least, very important, nucleus. Nucleus, I already said you, this is the controller of the cell. You call it as boss of the cell, right? And what they are having, number one, here we are discussing eukaryotic cell. That's why obviously it will have nuclear envelope. And inside what is there? The chromatin is there. Chromatin means in a form of thread. What is present here? Genetic material. I already said you, what is the genetic material here? DNA. And again here DNA is DS. What I said you, DS stands for double-stranded. Double-stranded DNA is a feature of all living organisms. Either they are prokaryotes or eukaryotes, right? Now, even here they need the medium. That medium you call it as nucleoplasm. Fine, already written here as well. Why I have written here? Because I want to say here, along with cytoplasm, right? Nucleoplasm together, you call it as protoplasm. Fine. What is inside the nucleus? Nucleoplasm. Outside, what is there? Cytoplasm. Together you call it as protoplasm. And what is this structure? Nucleolus. And nucleolus is a site for formation of what thing? Ribosomes. Keep it in your mind. Nucleolus factories are for what thing? Ribosomes. And ribosomes are the factories for the proteins, right? So that link you should be having. The ribosomes are actually present outside. Where? On RER and in cytoplasm. In mitochondria and chloroplast, 70S. But who is manufacturing that thing? That is the nucleolus, right? And last thing now, ribosome is mainly made up of rRNA and it is made up of proteins. rRNA stands for which RNA? Ribosomal RNA. I said you 3 types of RNA is there. Second type I discussed here right now, rRNA means ribosomal RNA and it is forming the ribosomes, right? So last slide, just concluding that, which is concluding means the last smaller structures which are still there, very small, which can be in plant cell as well as animal cell, that you call it as microbodies. 3 types are there, peroxisomes, spherosome and the glyoxysome. What is their function? That we will discuss in respective chapters where their role will come. But you should know these are micro, very small, but function is very precious. And these are again in vesicle, single membrane bound structures, right? They are many means they are many in number. They are many in number, but membrane bound. How many membrane bound? Single membrane bound. Fine. So this is all about the structures of the prokaryotes and eukaryotes. I hope so you understood. Just revise it, all the notes, and we will meet with a new chapter in our next session. Thank you very much.